I'm Hitoshi Shirakawa from Tohoku University. Thank you for such a nice introduction. I appreciate the organizers for this meeting and Dr. Yazawa for giving me the opportunity to make a presentation about our researches today. In today's symposium, we have three main topics. In the first session, I would like to show you an overview of the researchers in vitamin K2, and then introduce our research on vitamin K2. About 90 years ago, Denmark scientist Henrik Dam was studying the metabolism of cholesterol by feeding chicks with fat-free diet when the discovery was made. In this experiment, he discovered that the chicks developed bleeding symptoms and first speculated the absence of certain bleeding inhibiting or blood coagulating factors in the fat-free diet. He then focused his research on the substance before discovering vitamin K. Afterwards, Doisy, a scientist from the United States, and his team determined the structures of vitamin K1 and K2. Dam and Doisy were awarded the Nobel Prize in Medicine or Physiology in the year 1943 for their effort in vitamin K researches. The basic structure of vitamin K consists of a 2-methyl-1,4-naphthaquinone. Vitamin K1 from plants has a phytyl group side chain at the 3 position. While vitamin K2, mainly microorganism sourced, has several lengths of isoprene units as its side chains. These are the two types of naturally occurring vitamin K. On the other hand, compound without the side chain, which was used initially as synthetic products and is more recently utilized as a metabolic intermediate, is referred as vitamin K3. A common feature of vitamin K is that they act as cofactor of gamma-glutamyl carboxylase for the introduction of carboxyl group to the gamma position of certain glutamate residue on vitamin K-dependent proteins. This modified glutamate residue, called GLA residue, is found in blood coagulation factors. If this modification is not completed, blood coagulation will not occur. Other than blood coagulation factors, there are GLA residue, which is found in osteocalcin, matrix GLA, protein in bone, and blood vessels, and secreted proteins and some receptor-like proteins on cell surfaces, which are thought to be related to cell proliferations. Vitamin K functions are expressed via GLA modification through its responsive proteins. Here, research of ours where mice fed with vitamin K deficient diet for 1 week develops bleeding symptom is shown. In this experiment, this symptom is seen in germ-free mice that are lack of intestinal microorganisms, but not in normal mice. 動物にですね、このビタミン系欠乏食を与えてもですね、全くこういった、欠乏症っていうのはなかなか起こしづらくてですね、ここでは、特殊な動物というですか、腸内細菌は全く含んでいない、持たないような、無菌動物、無菌マウスを使いますと大体1週間くらいでこういった症状が出てきます。Here you can see rats being placed in a vinyl isolator where a germ-free environment is created. Consumption of a week of vitamin K deficient diet in these rats led to the prolongation of blood coagulation time that exceeded 5 minutes. このように血液凝固時間が5分経っても固まらないというふうなくらいまでですね、血液凝固が延長していくというふうなことが分かっています。In animals with intestinal flora, deficiency symptoms did not occur. The reason is that various intestinal microorganisms produce vitamin K2 of various side chain lengths. Here is a chromatogram of HPLC analysis of the colonic contents of the tested animal rats. We can detect menaquinone 6, 7, or up to 15. This data was from rat, but human intestine also has similar menaquinones. Some portion of this menaquinone may be absorbed by the host and thereby be responsible for part of the vitamin K source. When we measured the vitamin K contents in various organs in rats by HPLC analysis, vitamin K was not only observed only in bone, aorta and liver that synthesizes blood coagulation factors, but also in other tissues, including pancreas, brain, and testes. These animals were fed with diet containing vitamin K1 as a source of vitamin K, but not only vitamin K1, but also a significant amount of menaquinone 4 that has 4 isoprene units in side chain were found in organs too. A large quantity of vitamin K was found in the pancreas, but they are primarily menaquinone 4. While this shows the results obtained from rat, the similar can be observed in human, where ingested vitamin K1 will be converted into vitamin K2, particularly in the form of menaquinone 4 in the body. These observations have led to many researchers, including Japanese researchers, into the studies that revealed the mechanism of menaquinone 4 conversion and particular effects of menaquinone 4 other than vitamin Ks. As shown here, menaquinone 4 has been shown to be involved in cell differentiation, induction of apoptosis in various cells, and ligand activity for nuclear receptors. Here in this slide, a research in Japan is shown. In 2000, Japanese researchers published papers regarding the promotion of osteoclastogenesis and inhibition of osteoclastogenesis by menaquinone 4. Also, Japanese researchers revealed menaquinone 4 effects on the induction of apoptosis and autophagy in tumor cells. Within these studies, it has been shown that epoxide of menaquinone 4, which is normally an intermediate during metabolic reactions, interacts with cysteine residues of BAK, a protein that has a very unique ability to promote apoptosis, resulting in BAK-mediated apoptosis. Additionally, one of nuclear receptor superfamily, PXR, also known as SXR, is involved in regulation of gene expression of drug metabolism genes such as CAP3A4 and MDR1. Menaquinone 4 works as a ligand of PXR and directly modulates gene expression of drug metabolism genes. In relation to this discovery, Dr. Inoue and his group from the Tokyo Metropolitan Geriatric Hospital and Institute of Gerontology made a publication about the involvement of vitamin K2 in SXR-mediated bone homeostasis. The data are shown briefly here. The expression of SXR in osteoblasts was found. When menaquinone 4, which is noted as vitamin K2 here, was administered to these cells, the expression of these target genes of SXR was increased several-fold. Moreover, the same group found that activated SXR by vitamin K increased the expression of genes such as SCUSY and matrilin that are related to bone formation and collagen deposition in osteoblasts in a PXR-dependent manner. This further shows the significant effect of vitamin K on bone formation.
理論的にはビタミン K を上げているというふうなことも示してきて、よりビタミン K がですね、骨に対して大きな影響をですね、与えているというふうなことを示してきています。また私たちのグループではですね、
Our group has also found that activation of PXR in the liver by menaquinone-4 reduces the expression of genes such as CYP7A1 and 8B1, the rate-limiting enzymes for bile acid synthesis, in a PXR-dependent manner.
合成のリスト酵素であります CYP7A1 とか 8B1 といった遺伝子の発現をPXR 依存的に下げるということを私たちのグループでは見出しています。このようにですね、
In summary, although topics such as the induction of osteoblast differentiation, inhibition of osteoclast differentiation, and induction of apoptosis in osteoclasts could not be presented today, all studies related to the effect of menaquinone-4 on bone that are conducted in Japan have shown positive effects on bone formation. Menaquinone-4 is now prescribed as a treatment for osteoporosis.
骨粗鬆症の予防薬として、治療薬として販売されて、処方されています。
Other than this, menaquinone-4 can also induce autophagy in tumor cells, exhibit ligand activity for PXR and SXR, and promote neural differentiation, which I believe Dr. Sahara will talk about it later. These are all unique effects of menaquinone-4, which are shown in red, and all of them have been studied in Japanese research. In our group, we have shown novel effects of menaquinone-4, including activation of adenyl cyclase, upregulation of cyclic AMP in cells, activation of downstream protein kinase A and EPAC, and inhibition of NF-kappa B activation.
阻害というふうなことを示していきました。
More and more new effects of menaquinone-4 are gradually being understood. The Japanese researchers also have great contribution to reveal the mechanism of menaquinone-4 conversion from vitamin K1, that is the main form of vitamin K in our diet.
この問題に対しても日本の研究というのは非常に世界をリードしてまいりました。
Actually, the phenomenon of conversion of menaquinone-4 from vitamin K was already known 6 years ago. This was the conclusion that was made by Martis and his team in their paper about the conversion of vitamin K1 and menaquinone in 1964. They firstly proposed that intestinal bacteria firstly mediate the dissociation of the side chain of vitamin K1. Then the dissociated compound, which is a type of vitamin K3, will then be absorbed and converted to menaquinone-4. Later, Dr. Koma and his colleagues in our laboratory demonstrated that menaquinone-4 is produced even in germ-free animals, such as the one I mentioned earlier, where intestinal bacteria are not present and involved at all. This eventually led to the conclusion that vitamin K1 is converted into menaquinone-4 after absorption.
実際にビタミン K1 が吸収された後にメナキノン法へと変換していくことというふうなことが分かっています。
Professor Suhara and his colleagues that will be giving the speeches later about the identification of enzyme that is related to the conversion of menaquinone-4. This enzyme is known as UBIAD1, which is a type of prenyl transferase that introduces a side chain to vitamin K3. Here, the current understanding of this conversion is demonstrated. Vitamin K1, which is found in vegetables, and vitamin K2, which is found in fermented food primarily, fermented soybean, so-called natto, once being absorbed in the intestine, have the side chains dissociated, forming vitamin K3 in the process. This product is then transported to many tissues, where the side chain is introduced by UBIAD1.
UBIAD1 によって側鎖部分が導入されてメナキノン4ができるというふうなことまで分かっています。
In addition, our experiments with crude extracts from organs have also shown that this dissociation occurs not only in the gastrointestinal tract, but also other tissues such as the brain and kidneys. It is thought that the absorbed vitamin K is transported directly to the tissues and then converted into vitamin K3 by side chain dissociation factors, then converted into menaquinone-4 by UBI81.
変換されてくるのではないかというふうに考えています。で、動脈ですとか骨、それから血液、肝臓といったところでのKの機能というふうなものは十分に分かってはいるんですけれども、じゃあそれ以外の
Although the functions of vitamin K in the arteries, bones, blood, and liver are well understood, the role of vitamin K in other tissues, particularly menaquinone-4, is not yet well understood. For this reason, we firstly targeted the testes in our research. When rats were given feed supplemented with menaquinone-4 100 times more than normal amount, its blood testosterone concentration was increased. Moreover, the testosterone level in these testes were higher in the menaquinone-4 administered group.
また、精巣でのテストステロン含量も非常に高いもの、メナキノン4を食べさせたグループでは高いものでありました。
Since testosterone production in the testes is controlled by luteinizing hormone from the pituitary gland, we speculated that the amount of this hormone might be altered. However, the luteinizing hormone in the blood was found to be not affected by menaquinone-4 supplemented diet. Therefore, we presume that menaquinone-4 can directly modulate testosterone synthesis in testes. Next, the effects of menaquinone-4 were studied using Leydig cells, which is the testosterone-producing cells in the testes. In this experiment, we used two types of Leydig cells. In both cases, the testosterone secreted into the medium was increased by menaquinone-4 in dose-dependent manner. These results indicate that menaquinone-4 acts directly on the Leydig cells of testes to increase testosterone production. In the testes, cholesterol is used as a primary source for the production of testosterone, and multiple steps are involved toward the final product. It is believed that as a controlling mechanism, when cyclic AMP, protein kinase A, and upstream adenyl cyclase are stimulated and thereby activated within this axis, the rate-limiting enzymes STAR and CYP11A, which can convert cholesterol to pregnenolone, are stimulated. When we looked into how menaquinone-4 stimulates this signaling pathway, an inhibitor of protein kinase A, H89, completely blocked the testosterone production that was elevated by menaquinone-4. We also found that the elevation of cyclic AMP levels in menaquinone-4 treated cells. Although all data cannot be displayed here this time, we speculated that menaquinone-4 can firstly activate adenyl cyclase and increase intracellular cyclic AMP, then activate A kinase, and ultimately increase testosterone production. Here shows the mechanism that we expected, but we still do not know whether menaquinone-4 can directly activate adenyl cyclase, and whether there are some type of receptors that couple to adenyl cyclase. In today's super-aged and stressed society in Japan, age-related male hypogonadism, LOH syndrome, has become a major topic of concern. The decline in testosterone production can result in symptoms such as muscle weakness, sexual dysfunction, and depression. Additionally, there is a number of other symptoms such as metabolic syndrome and cognitive impairment that can occur when testosterone levels drop.
症状がありますけれども、この、低下といったところにですね、テストステロンのですね、テストステロン加齢による低下というところ
We believe that vitamin K supplementation can be used to reduce the decline in testosterone synthesis caused by aging, and therefore alleviate some of the related symptoms.
緩和できるのではないかというふうに考えています。
We also studied the possible functions of vitamin K in the pancreas by focusing on insulin secretion. This is a rat-derived cell that is known as INS1. It secretes insulin in response to glucose stimulation in the culture medium.
媒質にインスリンを分泌してくれるの細胞となります。で、この時にですね、
When the cells was treated with menaquinone-4, insulin secretion stimulated by glucose were enhanced by menaquinone-4 treatment.
このような形でメナキノン4をですね、入れること、共存させることによってインスリン分泌が増強することが分かってまいりました。また、先ほどのやつは
On the other hand, instead of the INS1 cells, we isolated islet Langerhans from mice pancreas and measured secreted insulin by glucose. In this study, we also observed that menaquinone-4 can increase insulin secretion in a dose-dependent manner.
メナキノン4の濃度依存的にインスリン分泌量が上がることが分かってまいりました。で、こちらの方はですね、
Here, an outline of the pathways that insulin is secreted from pancreatic beta cells by glucose stimulation is summarized. Glucose treatment increases intracellular ATP levels, which causes depolarization of the membrane. This then leads to an influx of calcium into the cell, leading to the translocation of the secretory granules of insulin to the cell membrane, and eventually insulin secretion. Other than these mechanisms, GLP-1 and GIP, substances that are found in gastrointestinal tract, can act as incretins. Both bind to the receptors on beta cells and upregulate cyclic AMP, and then activate downstream factors that laterally support and enhance insulin secretion. Considering our results on the testes, we speculated that menaquinone-4 has the function of increasing the production of cyclic AMP. Thereby, we conducted an analysis focusing on cyclic AMP. We found that even in INS1 cells, treatment with menaquinone-4 causes an increase of cyclic AMP in the cells. And about the downstream factors, we were interested in finding out whether protein kinase A, or EPAC, is affected by menaquinone-4, and hence conducted a study using inhibitors. The results showed that menaquinone-4 stimulates EPAC pathway rather than protein kinase, together with insulin secretion from the cells. All of the previous results were results from experimenting cells. For human and animal studies, the following results were found. Dr. Sakamoto from Hyogo College of Medicine did an experiment more than 20 years ago, where he divided students with various consumption levels of vitamin K into 3 groups, and measured their blood glucose and insulin levels after ingesting a glucose solution. The group with the lowest vitamin K intake were reported to have the tendency to have higher blood glucose levels and lower insulin concentrations. Also, a similar study performed on rats by the same research group found that when rats were fed with low vitamin K diet, they exhibited low insulin secretion in response to glucose administration. Their results were very similar to our cell-based research, and we think vitamin K, especially menaquinone-4, may be involved in the enhancement of insulin secretion from pancreas. In addition to vitamin K functions in liver, bone, aorta, vitamin K may have a testosterone-enhancing effect in the testes, from our studies, which may be a key in the prevention of LOH syndrome. And we also believe that vitamin K may be able to promote insulin secretion and hence be linked to the prevention of diabetes. Due to time constraints, I cannot share with you the researches related to brain, but our group have been doing researches on the brain recently, and we think that menaquinone 4 may be involved in the improvement of cognitive function, suggesting its potential in the prevention of cognitive decline. There are now more than 10 million people with osteoporosis in Japan. Osteoporosis is one of the causes of locomotive syndrome, and its exacerbation significantly reduces the quality of life of the elderly. As I have explained earlier, vitamin K2, more specifically menaquinone 4, is prescribed in the form of Grakke for the treatment of osteoporosis in Japan and East Asian countries too. In addition to its effects on bones, the role of this vitamin in the prevention of diabetes and dementia is gradually becoming clearer. I believe that vitamin K2 will become more and more important in the future as people seek to increase the healthy life expectancy and maintain the quality of life of elderly. This research was carried out in the Laboratory of Nutrition at the Graduate School of Agricultural Science, Tohoku University, under the guidance of the former professor, Dr. Michio Komai, and the previous professors before him, including Dr. Kimura and Dr. Furukawa. On the other hand, in the laboratory, Dr. Osaki, an assistant professor, has been leading the research on vitamin K. Moreover, Dr. Sato of JR Mills has always provided me with advices in many ways, especially on the increase of vitamin K and testosterone production in the testes. Nonetheless, I would also like to express my gratitude to the Japan Society for the Promotion of Science, the Tohoku Research Group for Lactic Acid Bacteria, the Mishima Kaiun Memorial Foundation, the Sugiyama Kokai, and other foundations for the support to this research. That is all for now. Thank you very much for your kind attention.